Okay, hello and good evening everyone, and welcome to myDNAhealth's live webinar on sugar metabolism covering genetics and epigenetics of disease prevention, which is scheduled to last about 45 minutes, including questions at the end, and hopefully it's going to be informative. Before you ask, it is being recorded and there will be a handout and we'll be sending that out hopefully by tomorrow. Okay, so just a little about our presenter this evening. Dr. Eve Pearce has a PhD in medicine and a BSc honors in medical biochemistry. She has over a decade of experience in genetics and medical research and has recently joined the myDNAhealth scientific genetics team, and we're delighted to have Eve on board. Following her PhD, Eve joined the University of Southampton's genetics lab as a research fellow, and during this time she published a number of research papers in medical journals and also trained medical students. Eve's passion for wellness and nutrition led her to complete an ION diploma in nutritional therapy, and currently she provides dietary clinical services for an NHS weight management program. Her clinical interests include genetics and epigenetics, nutritional biochemistry, and cancer research. I'm sure you'd all agree that Eve is highly qualified and has a lot of insights into epigenetics, genetics, nutrigenomics to share with us this evening. I'm certainly looking forward to listening to what Eve has to say. Okay, let's move on to the agenda. So this evening, Eve will be covering the following. She'll do a recap on sugar in the media. There has been a lot of media coverage around this topic. Blood sugar dysregulation, the importance of epigenetics, and looking at an integrated approach. Genetic variants, so she'll be discussing some of the SNPs for assessing sugar metabolism, including, very importantly, dietary impact. And then she brings it all together in a clinical case study. And at the very end, we'll then do Q&As. Throughout the session, you'll see on your left-hand column, there's a live chat. So by all means, you can post questions to everyone in the forum, or if you're feeling a little shy, you can just post it to the presenters, and then we can deal with those questions at the end. If you're having any technical or sound issues, please let us know by posting to the presenters in the live forum as well, and we'll be trying to deal with that through the session. Okay, so before we start, as we always do, there's a quick poll. So the question we want to ask here is, do you use nutrigenomic testing in your practice? Yes, no, or planning to? So we'll just keep it live there for a couple of minutes. So if you could start voting, thank you. Okay, great. So we've got 20% already using genetic testing, nutrigenomic, and 65% planning to. That's fantastic. With 10% not yet, but hopefully after these webinars we're running, you will be considering adding genetic testing to your practice. That's great. Thank you. So without further ado, I'm going to hand over to Eve, who will take you through the rest of the session, and then you'll hear from me at the end during the Q&A session again. Okay, Eve, thanks. Over to you.
Okay. Thank you, Bernie, for that lovely introduction. And it's interesting to see that many of you are using nutrigenomics or planning to in the near future. So hopefully we can help you with that sort of journey. So moving on, sugar's been in the news recently, and campaigns such as Jamie Oliver's Sugar Rush, who you can see here, have been very much in the public eye. Thanks to this campaign, the UK government has recently announced that a sugary drinks tax will be introduced in the UK in 2018. But is this enough, really? Is this enough effort? So the media are beginning to highlight a growing obesity epidemic and its complications such as diabetes due to poor lifestyle and dietary choices. But these views on the issue all too often draw on stereotypes. So you can see here some recent headlines that have been in the media, very much stereotypical. So they often present a simplified description of the problem, such as here a headline stating men have just a 1 in 20 chance of attaining a healthy body weight. What does that actually mean? This does not discuss current scientific evidence and the understanding and the difficulties of losing weight once overweight, obese, or even morbidly obese. And indeed, as we know, every person is different and requires an individualized approach. So this is obviously of a serious concern to the UK government. a problem which is having a profound long-term consequences on our health budget and to the wider economy. So, for example, cases of diabetes have soared by 60% in the last decade in the UK, and the immense pressure that's putting on our NHS. So that's about £14 billion being spent on diabetes and its complications by the NHS just last year. So we know that blood sugar is the monosaccharide glucose that circulates in the blood. This is obtained from digested food, and glucose is one of the body's main sources of fuel, providing cells with energy. And in the case of the brain, this is its only source of energy. So here we have an overview of the blood sugar roller coaster, which I'm sure you will all have seen before or something similar and be very familiar with. So as we know, excess intake of carbohydrates, in the case here an unbalanced breakfast, leads to diglycemia and a proportional increase of insulin secretion. So common symptoms of which include the fatigue, brain fog, fat storage, cravings for sweet foods. And this continued blood sugar dysregulation over periods of time can lead to a decrease in the sensitivity of body tissues to the metabolic action of insulin, and this is known as insulin resistance. So this change induces four biochemical transformations, leading to chronic elevations in blood glucose, insulin, very low density lipoproteins, and free fatty acids. So these biochemical variations lead to changes in physiology known as metabolic syndrome. This is a set of specific conditions and diseases that share the etiology due to excess refined carbohydrate consumption, which may lead to hyperinsulinemia, dyslipidemia, type 2 diabetes, and obesity. Evidence is also beginning to show that cancer risk through increased circulating levels of insulin-like growth factor 1 is important. And all these mechanisms really are driven by inflammation. So the underlying causes of metabolic syndrome are in our lifestyle and environmental changes, which is distinctive of civilization. Modern industrialized society encourages overeating, eating too many refined carbohydrates, and moving too little. Our ancestors worked very hard to find enough food in their natural environment to enable survival. Modern life has increased our lifespan, but the genetic survival mechanisms which evolved in our past through famine and hardship still have a very profound effect on us today. So this is where the importance of our genetics and epigenetics become apparent. Our genetic blueprint has evolved to enable us to metabolize sugar to ensure that our survival through periods of difficulty. Subtle changes in our DNA code, genetic variants known as single nucleotide polymorphisms or SNPs, can lead to impaired sugar metabolism, possibly leading to a propensity for obesity or metabolic syndrome. In addition, we know that changes to our diet and lifestyle, known as our epigenetics, have a profound effect on genetics we inherited and our underlying ability to metabolize sugar. So we have another quick poll here. What results matter most when you're advising a client? So is it SNP results, case and family history, lifestyle questionnaires, their symptoms, or are they all equally important? If you could just open the poll for me. Okay, we'll just let that run for a couple of minutes. Okay, so it looks like the majority are saying that it's all equally important, so you've got an overall picture of everything. Yep, that's really interesting. Lovely. If you could close the poll for me, please. Thank you. So just moving on from that, myDNAhealth use an integrated system which is designed to analyse the genetic variants, so the SNPs, and the epigenetic effect of our environment together. So today we're focusing on the genetic variants of sugar metabolism, which is part of the Weight and Wellness Management Panel. The SNPs we will be covering are TCF7L2, PPARG, FABP2, TNF Alpha, FTO, and MC4R. So that should give you a nice overview of the area without overwhelming. So firstly, we'll be looking at transcription factor 7, like 2, which is often abbreviated to TCF7L2. And this is a transcription factor protein which binds to DNA and affects the expression of genes and the amount of downstream proteins produced from this code. So the majority of genes consist of a region of coding DNA. This is the instruction to make a protein, which you can see here, which is preceded by a region of non-coding DNA known as a promoter. This promoter region allows the cell machinery to get in there and read the gene DNA and produce a protein. So it's really the on-off switch for the gene to be transcribed and translated into a protein. So as a transcription factor, TCF7L2 protein binds to the promoter regions of genes, allowing the cell machinery to produce proteins known to be involved in blood glucose homeostasis. So here we can see that TCF7L2 is bound to the promoter region of any number of genes important in sugar metabolism, and this allows the production of proteins key to glucose homeostasis. So this same mechanism is used to produce the TCF7L2 protein itself from the TCF7L2 gene. So here we have another transcription binding factor which is binding to the promoter region of the TCF7L2 gene, and this is allowing the production of the TCF7L2 protein. So that's producing the transcription factor. So this is repeated here on the left, but on the right, a SNP genetic variant produces a small change in the TCF7L2 gene coding. And when that cell machinery produces the TCF7L2 protein, this causes an alteration to the TCF7L2 protein structure itself. Now, this is a small alteration, but it affects the way that TCF7L2 acts as a transcription factor, which changes the downstream expression of genes involved in glucose homeostasis. So TCF7L2 as a transcription factor activates several other genes within the cell involved in receiving insulin to its receptor and sugar to the cell. The genetic variant, or T allele, is known to be associated with increased risk for insulin resistance and type 2 diabetes, and this is more so in the homozygote genotype than the heterozygote. So just to remind you, the homozygote indicates when an individual carries 2 copies of a genetic variant, and heterozygote carries 1 copy of a genetic variant and 1 copy of the original wild type. In this case, research has indicated that a reduction in refined carbohydrates and an increase in fibre may be beneficial for weight loss, and this dietary effect is greatest in those carrying 2 copies of the T allele. So let's move on to peroxisome proliferator activated receptor gamma 2, more commonly known as PPARG. So among its different functions, PPARG is also a transcription factor. This works together with another protein known as RXR, and together these 2 transcription factors affect the expression of many genes involved in apodogenesis. So the formation of fat cells, lipid metabolism, glucose homeostasis, and insulin sensitization. PPARG has been known as the thrifty gene, and it's crucial to survival in times of scarcity, and as a result, in times of plenty, this can contribute to obesity. So as in TCF7L2, the PPARG SNP genetic variant has been associated with a reduced ability to target crucial genes. This is due to an amino acid change at position 12 from a proline to an aniline. So interestingly, this change is associated with a protective effect against higher BMI and insulin sensitivity in response to higher free fatty acid levels. Therefore, this has a positive effect when consuming common plentiful Western diets by downregulating thrifty gene expression, and it is thought to offer some resistance to type 2 diabetes. So, in summary, those carrying a copy of the C allele, or the more common wild-type genotype, will benefit from a dietary decrease in saturated fat consumption and an increase in monounsaturated and polyunsaturated fats whilst addressing blood sugar dysregulation, and this is if they're consuming a Western-style diet. So let's move on to another gene variant tested in the Weight and Wellness panel. This is tumor necrosis factor alpha. Although many genes have been linked to metabolism, studies have indicated that TNF alpha, which is a cytokine released from macrophages during inflammation, has a crucial role in postprandial lipid and blood glucose levels. So a genetic variant this time located in the promoter of the TNF alpha gene has been linked with insulin resistance as this cytokine inhibits insulin. So just to remind you, the gene structure is composed of a non-coding promoter region to which transcription factors bind, which drives the production or translation of protein from the coding gene. So in this case, the genetic variant known as -308G to A causes a change to the promoter of the TNF alpha gene. The A variant improves the ability of transcription factors binding to the promoter in comparison to wild type G, and this increases the amount of TNF alpha protein which is produced. So you can see here we have the G and the A, and we're moving on. The cell machinery comes in. We have translation and transcription, and the increased levels of TNF alpha is associated with chronic low-grade inflammation, obesity risk, and insulin resistance in those with the A variant. So evidence suggests that maintaining an anti-inflammatory based diet, particularly high in omega-3 to saturated fat ratio, is very beneficial in maintaining glucose metabolism in those with the allele. So that's both the heterozygote and the homozygotes. So I'd like to now just look at two genes with variants associated with increased obesity risk. Now, both of these were identified through a relatively new research technique, which is known as genome-wide association studies, or GWA studies. Now, in this case, we can see DNA is extracted from an obese group and a non-obese matched control group. Using DNA microarray technology, hundreds of genetic variant SNPs are analysed in both groups and compared, and this is to identify SNPs which are associated with obesity. This technique has the power to pick up potentially important genes which would not have been identified through traditional known candidate association studies. So the first obesity-related gene identified in this way was called fat mass and obesity associated gene, also known as FTO. And this name really came about because they didn't really know at first what this gene did, so they just named it and then tried to figure out what happened. So a significant gene variation in FTO is thought to play a role in appetite regulation, which is associated with eating behaviours. The A allele has been associated with an increased BMI body fat percentage and waist measurement, leading to insulin resistance and diabetes. And this is especially prevalent when a high-fat diet is consumed, having the highest effect on the homozygote genotype. So that's those with 2 copies of the A. And this is thought to be present in 1 in 6 of the population. And they consume more fat and experience less satiety proportional to whether they are heterozygote or homozygote for this SNP. Interestingly, studies have shown that this obesity risk can be lessened by exercise, but that this effect has no effect in wild-type genotypes, so it's exclusively to those carrying the genetic variant. But the second gene variant identified using GWA studies is a melanocortin 4 receptor, which is a protein located in the hypothalamus region of the brain, and it's responsible for energy balance and food intake. In this case, the genetic alteration is a T to C change, and it's associated with increased risk of obesity due to increased energy intake. Those carrying the C allele show increased snacking and a larger meal behaviours. Research has showed that dietary modifications to increase protein intake in heterozygote and homozygote supports their blood sugar regulation and reduces the risk of obesity. So now we've covered those SNPs, and you can see there's a theme running through them. Let's move on to the effects of the environment on our genetic inheritance. So it's important to remember that a multifaceted approach is needed to restore health and prevent disease. So by using a number of health questionnaires, including cravings, toxicity, stress, burnout, sleep, weight, and cortisol, we can combine genetics and lifestyle epigenetics to design a personalized approach. And this is key, as it's important to treat the person and not just the genetic variant. So you may have heard treat the person and not the SNP before. Let's look at this approach using a case study. So this is the case of Mary, who has already been diagnosed with diabetes, that's type 2 diabetes. But if this was not the case, you would consider using functional tests to assess diabetes or pre-diabetes, as she's overweight over the age of 40. So other factors to consider are family history, blood pressure, ethnicity, and symptoms such as fatigue and excess urination. So functional tests which are routinely applied to diabetic diagnosis and management in the UK are a fasting plasma glucose test, glycated hemoglobin test, and oral glucose tolerance test. This slide just shows you the current NICE guideline test ranges, and these are all routinely available through GP, so you should be able to easily get hold of these results. Okay, so looking at the results of Mary's nutrigenomics testing, we can see that there are severe problems with her sugar metabolism, and this is indicated on her summary picture of her results, which you would get in any report. So let's break this down into more detail, and it will give us some more information when developing a personalised strategy. So Mary has a wild-type genotype for the MC4R, showing that dietary changes will have no effect in relation to this genetic variant. But she has one allele copy of the genetic variant for FTO, indicating possible increases in appetite regulation and weight gain, and that moderate exercise would have a beneficial effect to her. So interestingly, when we look at Mary's questionnaire results, so this is the epigenetics side, for food cravings and hunger, these correlate with her genetics, as she is classed as a severity. So using the nutrigenomics and the epigenetics indicates that Mary is not regulating her gene expression, and she's increasing her risk in this area. So due to her FTO genetic variant, recommending moderate exercise and keeping portion control in check would be important and beneficial. So let's move on to the remaining 4 genetic variants. These are important in sugar metabolism. So we can see that Mary has a wild-type genotype for fatty acid binding protein 2 and TNF alpha, indicating that dietary strategies for these variants would have little effect. However, she has a wild-type genotype for the PPARG and a homozygote for TCF7L2. And as we saw before, these 2 genetic SNPs indicate she overabsorbs sugar and struggles to metabolize her sugars correctly, and dietary advice in this area would be beneficial. So the PPARG, if we remember back, the wild type, the CC, that was the thrifty gene, so it would be a beneficial effect to have dietary effect there. So Mary's questionnaire results for stress and burnout indicate moderate levels in these areas. So this indicates she's not managing her reduced genetic capacity to metabolize sugars correctly, leading to blood sugar dysregulation. And this is further evidenced by her BMI, waist to hip ratio, and type 2 diabetes. So taking Mary's combined results into account, nutrition and lifestyle advice should centre around eliminating all sugars and refined carbohydrates, eliminating seed and vegetable oils, some moderate exercise, that's important for that FTO variant, as we saw the research has really highlighted that as a protective effect. Techniques to reduce anxiety and stress, improving her sleep pattern, addressing her blood sugar dysregulation, so her type 2 diabetes, a healthy diet to include nutrients to support her sugar metabolism, and you could consider supplementing to support her sugar metabolism. So another quick poll. When it comes to advising about low sugar eating, what are you currently using? So we have glycemic index, glycemic load, sugar content, carbohydrate content, or none of the above. If you could just open the poll for me. Thank you. Okay. It looks like a high proportion are using glycemic load. Thank you. Yeah, so most people seem to be using glycemic load. That's the technique that I would use myself. It works very well in clinic. I found it works very well with NHS patients as well. It's quite easy to understand. So this would be appropriate for Mary to help support her sugar metabolism, help support her genetic variants, and really sort of take into account her lifestyle factors, her epigenetics. Okay, if you could just close the poll for me. There we go, lovely. So dietary advice for Mary could be centered around the myDNAhealth guidelines and in this case the low sugar food plans. These are all available as a resource that you can tap into on the practitioner portal and as additional information for your client. You can also ring and speak to the team for any extra advice as well. So there's plenty of support there for when implementing genetics, nutrigenomic testing, and including lifestyle epigenetics as well for your protocols for your clients. So just getting near to the end now, it's important to highlight that we really check progress. So Mary should retake her food craving, stress and burnout questionnaires after 2 weeks, and then again monthly as required. And it's also important that Mary has follow-up appointments if part of her strategy to check her ongoing supplement needs, look at how she's getting on. This could all be, you know, to see how well she's managing her lifestyle factors. So as a practitioner, how do you get started? Most of you are thinking about starting nutrigenomics soon, so hopefully this slide will be of use. So by usefully putting nutrigenomics into your practice, you need to treat the person and not the SNP. Really learn that gene function so you can understand what's happening with the genetic variant, because they work in many different ways, so it has to be the one that is appropriate for the nutrigenomics testing. Gain experience by practicing, talking to people, you know, just as much as you can, really get involved. Make sure that your genetic testing company provides you with excellent support. If your client is healthy, then support through diet and lifestyle, or if they're symptomatic, further functional testing may be needed. So we talked about the functional testing for diabetes earlier. If you decide to start supplementing, always go very low and very slow so that you sort of negating the possibility of undesirable effects, which we all know can happen in unexpected ways. And always follow up your client to make sure that what you've put in place is appropriate and is working, because if it's not working, you need to think again and change your strategy. Okay, so our key learnings are the SNPs involved in sugar metabolism and their function. We've looked at the benefits of an integrated nutrigenomic and epigenetic approach. And we've also looked at what to look at when giving nutrigenomic advice to your clients and how to track their progress. Okay, so I'm going to hand back over to Bernie now for any questions.
Oh, that's great. Thanks, Eve. So we've had a number of questions posted in the chat forum this evening. So let's go and tackle them all. So the first one is, what are the main genes for diabetes?
Okay, so we talked earlier about GWA studies. There's been many for diabetes and looking for candidate genes. So I'm assuming that this is type 2 diabetes that we're talking about. And they have identified over 500 genes that are associated with type 2 diabetes. So that's a considerable amount. And interestingly, I believe that obesity is higher up at 900 current genes. So this is a huge area, but some of the ones that we've talked about today are very, very relevant for diabetes, as we've discussed.
Great, thanks, Eve. Just to add to that as well, is that we keep our eye close to research on new SNPs relating to the areas that we actually assess, and provided that there's clinical evidence, it's reliable, it's relevant, and very importantly, it has that epigenetic aspect. So there's something that we can do. So it's the lifestyle, dietary, nutritional modifications. Then our clinician team will then develop and add those particular SNPs into the different panels. Okay, so the next question, do the genetic markers change over time in relation to age and lifestyle?
Okay, so your genetic code, your DNA code doesn't change. That's what you were given at birth, and that sticks with you throughout your life on the whole. But your epigenetic markers very much can be changed by age and lifestyle. So that's the important bit that we can regulate. And as we know, changes in nutrition and diet can have a profound effect on our health.
Thanks, Eve. So we're going back to the integrated approach that you were referring to earlier, looking at the environment, looking at the case history, any family history as well. Sorry, I think, Eve, you'd muted yourself. Okay, sorry, Eve. All right. Okay, and the next question, is it only glucose that the brain uses for energy? Does it also use ketones?
Okay, so if you're thinking in relation to a low carb diet, the body can utilize ketones in order to produce energy for the brain. So I think that's the area that you're trying to come into. But traditionally, glucose is the only brain energy that is known. But obviously, there's mechanisms and biochemical mechanisms in place that we can utilize ketones as a source of energy, but it's not as efficient.
Okay, great, thanks. Okay, if the PPL SNP result CC, will this person be sensitive to sugar and excess carbs? And are there any fats to be avoided as well?
So as we covered in the presentation, in this case, the CC SNP is the wild type, but that indicates that dietary changes are necessary because this was the thrifty gene. So decreasing saturated fats would be beneficial, decreasing your trans fats, hydrofats, all of that area, increasing your polyunsaturated, so omega-3 would be important here, but obviously in association with that low sugar dietary lifestyle modification.
Okay, what about your butters and coconut oils?
Okay, so the butters and coconut oils would be preferable to using sort of your trans or hydrogenated fats, but just in a smaller amount, so the least processed would be best here.
Okay, so it's about portion control.
Very much so, yes. Yeah, portion control.
Okay, great. All right, next question. Can gene tests provide clarity to my client's LDL levels, which has for years been slightly above the upper bound levels, no matter what diet and lifestyle she is following?
Okay, so this is quite an interesting area. You can certainly look at gene variant SNPs to get an idea of cholesterol metabolism and how that's being sort of metabolized by the body. So you can come at it from two sort of areas. You can look at the genetics, so you could look at some of the SNPs that we've talked about today. They do have an effect on lipid metabolism, but also some of the more directly associated cholesterol genetic variants such as the ApoE, ApoA2, all the apolipoprotein sort of family, very involved in lipoprotein metabolism and important for cholesterol metabolism there. Or you can come at it from the epigenetics point of view and look at factors such as things we know of like iodine, copper deficiency, and selenium, which would be important from a diet and nutrition sort of point of view, and bring that together when designing a protocol for your client.
Oh, thanks, Eve. That was quite a comprehensive answer. Okay, I think we've got time for one more. So my client has been eating a very low-carb diet for over a year and has always had normal blood sugar, but when tested recently, her blood sugar levels were above normal. How is this possible?
So this is quite interesting because it can be quite normal for people on a low carb diet to experience a rise in their blood glucose level. This can happen immediately or later down the line. So in this case, I don't think it's a first phase insulin secretion change. I think it's where the body has adapted, so to speak, to a low carb lifestyle. And what happens is your muscles will become insulin resistant, which is a physiological change as opposed to insulin resistance that we know as for metabolic syndrome. So I think at this point it would be important to really check your client's stress levels. So epigenetics could be having an impact here. As we know, cortisol upregulates gluconeogenesis, so that's breaking down protein to sugars. You could check for signs of stress, such as acne, fatigue, irritability, sleep issues, and really maybe consider that muscle mass is become insulin resistant. And this is a mechanism that protects the glucose, as I did mention before, for the brain, because that's its key source of energy.
Okay, great. Thanks, Eve. Okay, I'm just looking at the time. It's 7:10, so I have another question if I can pose it. Sorry, and then that will probably be the last question then. My client is insulin resistant with ME symptoms, so would a low sugar, healthy fat eating lifestyle be beneficial?
Okay, so yes, I guess to balance energy levels, that would be really important. For the ME, you're thinking about supporting mitochondria, so it's important to have nice steady sugar levels for that. This would also help with their oxidative stress. So anti-inflammatories would be important to put in here. So you could build your whole protocol around including that.
Oh, that's great. Thanks, Eve. Okay, I think we've got to the end of the session. If anyone needs additional help or advice, you've got the practitioner support team details on your screen. You can email us at practitioner@myDNAhealth.co.uk, or you can call us and we're here to help. Again, it doesn't matter what the question is. There's no stupid questions, so please get in touch with us. Okay, so Eve, thank you so much for your time in preparing this informative webinar. I've certainly enjoyed it. I think it's been very insightful as well. If you'd like to hear Eve again, she's presenting the next webinar on genetics and epigenetics of autoimmunity. How can nutrigenomic testing be useful in practice? And that's on the 11th of October, and we'll be sending out details in due course. So I think that's it for me. Thank you, everyone, for attending on this Tuesday evening. I hope you enjoyed it. I hope you found it informative, and have a good evening. Good night.